As I said, thanks for the organizers, thanks to the people who voted for this presentation. This presentation is going to be related to this topic. And welcome to the session. I will be explaining something about using votes to combine rankings. First of all, about me. I have background in mathematics. I work as a postdoc researcher in a biomedical genomics group, which in principle you would think it's not quite related to math. Our focus is on mutational processes and tumor evolution. We ask questions about how cancer evolves, how cancer develops, and this has a lot to do with evolution and mutation of the genome. I'm interested in modeling, statistics, and programming, and my long-term goal in life is to do once in a lifetime advent of code in Haskell. Let's see if I'm succeeding. But first of all, before going to the main topic of the talk, I want to tell you a story. 2 years ago, my girlfriend joined a very particular community of neighbors. It's a community of neighbors of this building. By that time, the building was crappy. It had serious issues. The front of the building was falling off to pieces to the street. It was really, really dangerous. The city council was threatening the community to shut the building completely, not allow anyone to live there. So the community faced several challenges. The meetings were very harsh, and people fight to each other constantly. So very violent politics. So a meeting in this community of neighbors would be more or less like that. I'm simplifying the story quite a bit, but let's say the meeting gathers and there are three main priorities on stake. They have to decide what's the priority, whether painting the corridor, fixing the front of the building, or doing the 10-year periodic pipe maintenance. So each one of the participants would have an opinion, okay, either pipe maintenance or fix the front. These are the priorities of this guy. This guy has a very clear idea that fixing the front is the top priority. And this one says, okay, paint the corridor or fix the front, paint the corridor. I don't really care, I hate these meetings. Most of the participants will fit in this psychological mood. So in the end, we can picture all the opinions of the participants in this meeting as an ordered set of options. These options, these relationships between the options may be represented as an equal, indicating that they don't really care whether one or the other, or with a greater than, indicating that this option is greater than in terms of priority to the next one. So using a normal voting method as the one that I will explain in a while, we may merge all these opinions to produce a consensus opinion of what's the priority. So we have several rankings in which we allow ties, and these rankings can be merged into a consensus ranking. But by that time, I saw my girlfriend was suffering a lot. She was under considerable pressure, and it was not very clear that the final decision was going to be fixing the front of the building. So what would happen in a normal situation is that people would cast their votes, some decision would be passed, and this decision will ultimately have an impact on the community, on the people walking in the streets. If another meeting had to take place in 1 year afterwards, then all the people will have the same voting rights, regardless of whether this was a wise decision or not. People who voted for not fixing the front of the building, having one person killed in the streets, would have the same voting rights. So I had a fantasy, which is perhaps not practically feasible, which is summarized in this schema, which is, okay, let's consider this, it's a thought experiment. let's suppose that votes are not immutable. We can change votes depending on whether the impact after casting the votes and passing the decision is good or bad. And then we can update the voting rights of the participants accordingly. And then the next voting will be based on the updated voting rights. In such a way that, for instance, if the decision passed turns out to be beneficial overall, I would not enter very in detail what beneficial may be in this case. People will have new voting rights, or voting rights may be transferred from one people to another. But this idea is really crazy in terms of practical feasibility in real-life social choice, because normally the definition of choices is not as straightforward. The feasibility of impact evaluation is mostly unfeasible most of the time. It's very difficult to isolate one consequence for the several factors that have intervened and that make that some outcome appears. And also the ethics of mutable voting rights is delicate. But I work in cancer genomics, so I was thinking this by the time I was trying to rank some things that I will call cancer driver genes. So I thought, maybe in social choice with real people, this is very difficult to apply, but maybe with machines, that also produce rankings and cast opinions, this may be feasible. So the overall problem I was facing by that time was the discovery of genes that drive tumor evolution. We, and myself and other people in the lab were involved in a project where we were bound to analyze a lot of information regarding the genome of patients who have cancer and trying to identify signals of positive selection in genes that may be critical for tumor evolution. But I will go step by step. What I mean by genomics is the study of the genome. The genome is the operating system by means of which living organisms function. Cells resort to genomes to do almost everything. Genomes are very particular operating systems written in a suitable extension of a very simple alphabet, which has just 4 letters, as many of you may know, which represent the basic building blocks of the very long stretch molecule, double-stranded DNA molecule. So in the picture, you can see how this operating system is physically organized in a chromosome. The chromosome contains plenty of these letters. It's a very long word. Among the parts which have meaning in this very long stretch of letters, the minimal functional unit is the gene. Genes encode for particular functions in the context of this very huge operating system. And we can think of genes as the minimal functional unit, or the minimal unit of these programs that has some meaning by itself. So we study genes in connection with cancer. Genomes learn to cooperate by evolution in multicellular organisms. Multicellular organisms are those which have different tissues made of different types of cells, and each cell is run by a different version of the same genome. Let's say it's the genome tuned to perform specific parts of the job. But when this balance, this cooperation breaks, then some cell populations may want to take over everything, and they break the balance of the healthy organism. How these cells break the balance? They take advantage of changes that appear just by chance over time. Genomes change. This is a matter of fact. We are born with cells with genomes, and these genomes, depending on the exposures, depending on what we eat, what we breathe, what we think, perhaps, we tend to accumulate mutations over time. Most of these mutations do not have any effect. They do not have any impact that we can measure. But some of these mutations are relevant because confer the cell with the ability to wanting to take over. So tumor evolves after one lucky or unlucky mutation is produced in one cell. This cell may change its program, may want to take over everything, and may start to grow and expand, dividing many times, creating a lot of progeny. And each time this progeny acquires a new mutation, a new change in the genome that's beneficial for it to compete with the others, then a new subpopulation may emerge. So this is a process that may take many, many years. And what we have is, after all these years of variation and selection, we have a very fine-tuned machinery hacked to compete against all the cells in the surroundings. Some genes are particularly sensitive to these changes, which I may call driver changes. These are some of these genes. And one thing that people working in this field has asked many times is, okay, these genes, the genes in this list, for instance, are very sensitive to, after acquiring one change by chance, conferring the cell with this new program of wanting to conquer everything. So one question that's been posed long ago, and it's still a matter of study, is after looking at the genomes of patients that are sick with cancer, can we retrospectively, can we reconstruct how the evolution of the tumor we are seeing was? Which genes were critical in order to this tumor to develop? So one can see these genome modifications as slight variations of the program. If we do very big modifications, the program arrests functioning and the cell dies. So we are talking about subtle changes in some cases. Using this example, we may see in the genome substitutions, we may see deletions of one letter, we may see copies of words, we may see one part of the genome translocated to another part where it was not originally. Sometimes the meaning changes very slightly. Sometimes the meaning changes significantly. So there has been some statistical methods proposed in order to analyze samples of patients and try to reconstruct how this evolution was. Reconstruct the evolution means producing a ranking of the genes that have most signals of positive selection. Normally, these ranking genes are produced after computing what in statistics is known as p-value. Probably many of you know about them, but I will very briefly refresh what a p-value means. Suppose we have a model, a statistical model, that has an opinion of how mutations appear in the genome or in a part of the genome. Suppose that the model expects to see the picture in the left, and it sees the picture on the right. We are surprising the method, in a sense. The method is embarrassed after observing the real data. This degree of embarrassment of the method or the statistical model is what we try to measure with the p-values. So the lower the p-value, the higher the degree of embarrassment of the statistical model. And this is how we try to identify these driver genes. So many statistical methods of this kind have been developed and proposed and presented in the literature, but they don't quite agree most of the times. They partially agree, but many times they don't agree or they don't agree at all. So we want to make sense out of all these rankings. So each method has an opinion of which genes were priority genes in the history of the tumor. And we may want to combine these rankings. So as I explained before, these rankings are given by p-values, and there are some standard methods to merge p-values when we have independent samples or independent experiments. So there has been these methods proposed for a while, Fisher, Stauffer, Brown, etc., which given p-values gives you a new p-value. So if we are capable to produce a combined p-value, then why is it problematic to combine these rankings? Because of the inconsistency of the rankings. Most often we are facing the consensus list produced by a combination of p-values, and this list doesn't make sense in terms of the consistency of the ranking. A gene that was not preferential for almost all the methods may appear ranked highly. And this is in part due to the fact that different methods use different scales of embarrassment. So a p-value may be low in all the methods, but low may be 0.01, 10 to the minus 10, 10 to the minus 100. So we wanted to correct for these rankings relationships that we observe in the individual methods. We want a consistent ranking that also gives us a systematic allocation of credibility, and that remains as interpretable and statistically sound as possible. So for solving the first step, the ranking consistency, we were thinking about, okay, let the methods vote. Each method produces a ranking of preferential genes. Let's make them vote. And for this, we choose to do, this voting can be made in many different ways. We chose the Schulze voting, which I will briefly explain. So this method is meant to take several rankings and produce a consensus ranking. This method of voting is particular because it's Condorcet. Condorcet is a property of voting methods. Not all voting methods have this property, but it's the kind of idea that we were intuitively looking for when we were asking for ranking consistency. So Condorcet means essentially that if we forget about all the candidate genes except 2, if one of the genes is preferred more times than the other, in the final ranking, this gene will be higher ranked than the other. This basic property is what we mean by Condorcet. So among the many, many advantages that this voting method has, it has this property of the Condorcet. Yet it remains fast to compute. It's a method that is cubic in the number of candidates. So it's, in principle, it's not a big deal of complexity. I will present you very briefly an example. So suppose we have 4 voters and 5 candidates. Each voter would produce a ballot in which it indicates in which ranking position he sees each of the candidates. There are some technical conditions that have to be fulfilled in order to have a valid ranking. So with these ballots, we would produce what's known as a weight matrix. Weight matrices basically reflect the preferences, how many voters prefer one candidate over another. We can reflect all this information in this matrix. Here you have an example. I will not go very much longer. After producing this matrix, we can represent it as a complete graph with weights in each edge. And by performing some max flow search of all the possible paths, of all the possible pairs of candidates, ordered pairs of candidates, Schulze's method manages to give you a final ranking which has the Condorcet property, which can be computed in cubic time in the number of candidates. So I will not go into the details of this algorithm. It's very nicely described in Schulze's paper. So just for you to have a very superficial idea of what kind of algorithms underlie. So the first step is already accomplished. How do we infer the credibility that each method has? Okay, we want to give higher voting rights to methods that contribute more to a better outcome. But what's a better outcome, after all? Aren't we entering into a kind of circular argument? A better outcome for us would be based on a consensus list, which is called the cancer gene census, which is a list of genes that have been experimentally validated to have an association, and strong association, with cancer. So whenever we see in the consensus ranking that the cancer gene census genes are at the top, or appear more often at the top, we will give a high score to this consensus ranking. We may define many different enrichment scores based on the frequency of CGC genes that appear on the top of the consensus ranking. We just are using a few of the most simple ones one can conceive. And now comes the critical point. I've not talked yet about voting rights, but we can pretend that the voting rights can change between these methods. So we can rebrand the algorithm of Schulze to make the different voters have different voting rights just by scaling up or down the preferences we have considered in the weight matrix according to a weight factor that would be unique for each method that produces a ranking of genes. So if we make them vote given a list of or an array of voting rights, a different result would be produced. So this gives us a function that connects the voting rights with a score that tells us how good is this final ranking in terms of consensus and cancer gene census terms. So we have reformulated the allocation of credibility as an optimization problem. We want to have the CGC score as high as possible by tuning the voting rights. So the voting rights we will choose will be the ones that make the function score, the CGC score, as high as possible. In practice, when we do this, the implementation of this method, suppose we have a family of methods. I'm not going to enter in the particulars. We would see if we represent the enrichment in CGC genes in the ranking in the first 20 positions, in the first 40, in the first 60 in our ranking of genes, we would see that this enrichment decays a little bit. But doing it with Schulze, we manage, and this optimization algorithm, we manage to accomplish something slightly better than the state of the art. So what's left? We were meant to select genes, and now we have new pieces of evidence. We have each gene ranked by the ranking combination given by the optimal voting rights we have been able to allocate in each method. And now, with these weights, we can as well perform a p-value combination a little bit with more accuracy, or using the new information about the credibility that each method may have. So with these two pieces of information, we can create a composite rule which says, for instance, okay, we will select the top genes of the consensus ranking as long as all the genes have a very low p-value, below a p-value threshold that we arbitrarily impose. And this is the end of the story. So how did we implement this idea? It was really instrumental using the NumPy and the Python in order to make the voting process very fast. The voting process has to be very fast because we want to optimize a function that's based on doing this voting process many, many times. So it's time we do this is time consuming, and if we do it many, many times, and if we use optimization problems that do not care about the number of evaluations of the objective function, it may be very expensive. So we sorted out this using the in-house knowledge about Python, and how to statically compile chunks of Python code. In order to represent and understand the graph of the method, it was very useful. This package, the network X, in my opinion, it's very well documented. The learning curve is not very high, and it's a quite complete package. The core part of the algorithm, as I explained before, is the one in which we do a max flow search of all the possible paths between all the possible pairs of candidates. This is how the Python code looks like after doing the proper translation of the algorithm that it's customarily used in this problem, which is Floyd's algorithm. By the way, this picture is the entire paper of Floyd. It's just that. In order to carry out the implementation, we mostly resorted to scipy and the array of different methods that it has. We did some attempts doing fancy things like augmented Lagrangian particle swarm optimization, Bayesian optimization, in order to try to reduce the number of evaluations we do in the optimization process, but it revealed to be an overkill. We got better results doing a very simple grid search in the simplex, where we look for the weight combinations, and after we have some good candidates, do a local search, hill climbing search with a little bit of temperature, perhaps doing a Bayesian hopping to introduce a bit of stochasticity, but the simplest approach turned out to be the best so far. All this is packaged in something that will release soon, in which you can carry out your own experiments, and all the functions that will be in this package are in turn mounted in a bigger project that we have in the group, in which we are bound to analyze very large cohorts of mutations in different types of cancer. So what you will be seeing here in this package, it's a condensed version of all the tools we used in this project, which is Introduzione is the project I was referring to. Is a website in which you can interrogate about drivers of cancer, about rankings of genes that have to do with different signals of positive selection. And this is one of the important projects we are having in our group. So what I explained so far is just one tiny, not so tiny step in the pipeline where we analyze these genes. So in summary, we have gone from neighbor politics, which is in my opinion the harshest form of politics on earth, to driver discovery. Making methods cast their opinions, combining these opinions using a voting system, which we decided it should be Schulze, and letting these rankings be evaluated using the CGC enrichment criteria. There are different optimization strategies we explore in order to do this search and come up with different weightings or voting rights we can allocate to each method. And this picture basically summarizes this part of a bigger project that is Introduzione. So the most important part of the talk is the credit and the thank you. This work I've presented here was done in close collaboration with Fran, which is seated over there. And it's part of a bigger project contributed by many people. All these ideas wouldn't be possible without the cooperation of all these people that contribute to this project. And special thanks to Loris, because he's not only part of the project, he dragged all us to this beautiful conference in this fantastic venue. So thank you, Loris, for this idea of yours. And that's it. I'm very happy to take questions, either here with the audience or if we can hang out outside. Thank you very much.
Thank you. So, any question? Okay.
I have a question about the scalability of the algorithm with the number of genes. So maybe I missed something, so let me know if I say some mistakes. So when you compute the algorithm to compare the preferences and then aggregate it, you basically count the preferences for each pair of genes, right?
Yes.
So what is the number of genes, and doesn't it explode computationally?
Yeah, the human genome has about 30,000 genes. Correct me, the biologists, please, if I'm mistaken. This is the scale of the number of genes we have. We have to take into consideration that we don't see mutations in most of the genes. So this amount of genes further reduced to a few thousands or a few hundreds in some cases. So this is the size of the problem. This size doesn't basically change, provided we don't change the genome. If we try to reproduce this analysis in another species with a different number of genes of different types of cancer, we may observe tenfold up or down changes in the number of genes, perhaps. Some species have much more genes than in the human genome.
So when you have to compute the metrics for each possible pair, it's still feasible?
Yeah, it's feasible. It's feasible. The problem is doing this iteratively many times when it comes to the optimization, if the optimization carries out a lot of evaluations. But we have to, we kind of sorted this out by using a little bit more efficient implementation using this Python chunk.
Hi, thanks for the interesting talk. I have a couple of fast questions.
Yeah.
The first is, if you reward the classifiers, the rankers, that consistently find the known drivers, don't you risk to reduce the probability to find new drivers?
That's a very good question. We are not 100% happy of the reward system we're using, but it's the best to our knowledge we can resort to. So in the list of genes we have produced after analyzing using this schema, many well-known genes are recovered, and it's true, it's a work in progress. It's true that not many new genes appear. It's an open question. What's the number of real driver genes in human cancer? We don't know how many. It's a very controversial issue among biologists. But I would say that some candidates that are not classified as CGC, that are appearing top ranked in our rankings, have some good signs or good chances to be relevant in some way or another, just by looking to other features that are not considered in this experiment. But this is a manually curated analysis that has to be done after we've produced the final rankings.
And the second question is, have you studied the weights given to the rankers? What happens if you remove the lower weighted classifiers?
I didn't understand, sorry. I did not get the question.
This method gives different weights to the different rankers. Then, is there a huge variability among the weights, and what happens if you remove the weaker rankers?
Yeah, we did this analysis. And we found out that most of the methods we were using, which in some sense were already validated by other means, most of the cases, all of them have a positive contribution to the score. In very few cases, the addition of a new method makes the result worse. It's a very good question. This is something we went through in the downstream analysis of the results of these experiments.
Thank you.
Okay, I have a question. Thanks for the talk. How cross-disciplinary is your package? In particular, does it apply to any statistical problem where you have to mix p-values associated to different rankings?
In principle, it's a general solution as long as you produce rankings that are the result of sorting by p-values, and as long as the scale of the problem is feasible. So one thing is the scale of the problem. The scale is very particular. Not all the problems may have this scale in terms of the number of candidates. But it's a method intended to tackle other problems. I cannot imagine other problems, just the neighborhood community problem and this one. But I'm convinced this is something that comes up recurrently in other contexts, even though I cannot tell you very particularly which other problems this would fit.
Yeah, I was asking because I work in economics, so
yeah,
we have some problems like those.
Thank you. Perhaps we can hang out later.
Hi, thanks for the technique. I wonder how the research for the weighting those classifiers looks like. I mean, how samples are getting from patients, I assume. So how the process looks like.
Yeah, it's a good question and it's a long answer. Perhaps I don't have the time to explain it properly, so we can discuss it later. But it's basically during the clinical process of dealing with the disease, some biopsies are produced. So big consortia of researchers collect these biopsies and stick to a uniform procedure to do the sequencing of these biopsies. So once we have done the sequencing of all these biopsies in a uniform way, we do what's called as a mutation calling. We filter this mutation calling by plenty of confounders that can come along the way, and we finally get to a table with samples and mutations of different kinds. Like the kinds of mutations I presented before, what's the context of each mutations in terms of the letters that come 5' and 3', and we try to dissect all these mutations, and all these statistical methods feed on this information.
Okay.
That would be more or less a quick summary of how it works.
Thank you.